Good afternoon, everyone. I believe mine is the last talk on this stage today. I want to thank the organizers for letting me be here, especially Dr. Srijit and Dr. Divya. I think I had a really wonderful time listening to all the talks, and although people say zebrafish is a small organism, we feel really bad when we kill so many organisms. So with that, I'll go to my slides. So what you're seeing on the left here is a zebrafish head, and I work on long non-coding RNA in the field of vessel integrity using zebrafish as a model. So what are long non-coding RNAs? These are transcripts which have length longer than 200 nucleotides, and they can be transcribed by RNA pol2, and their ORF is less than 100 amino acid. A hallmark of LNC RNA is to be expressed at a very lower quantity than a protein coding gene, and while protein coding gene is ubiquitous across different tissues, a LNC RNA would only be expressed in 1 or 2 tissues. And another hallmark is a poor sequence conservation, which makes it difficult to study LNC RNAs. However, they are conserved at their functional level through having conserved structural domains. Now, I am glad that we had a lot of talks on zebrafish and on angiogenesis, which makes it easier for me because they have all become primers. Thank you especially to Dr. Idris. Today he made it very, very easy. He explained a lot of things. So, vascular development in zebrafish is really conserved. And what you're seeing here in green is the development of the vessels in zebrafish. So this is the ventral mesoderm from where the precursors of endothelial cells arise. They migrate to the midline of the embryo. They form the primary vessels. This is called as vasculogenesis. And all of this happens within like 1 day. And from the first day, then angiogenesis happens, which is the formation of the secondary vessels. Now, at the cellular stages, this is how all the stages happen. Now, all the protein coding genes have been studied for the 100 years to understand how all of this is regulated. Recently, non-coding genes have been implicated, and even LNC RNAs have been implicated. And one such study is from our lab, in which we used a double transgenic zebrafish. The endothelial cells are labeled, and we did poly-RNA sequencing, and using a custom-built pipeline, we identified LNC RNAs. And 6 of them we call them as wheels, which we use for functional validation. And the main study is published in the EMBO journal. We had the cover pic as well, and this is the study if anybody wants to go back to it. So for my study, I picked a candidate called wheel6 because it is syntonically conserved. So what is syntony? When you look at these different organisms, if genes are placed side by side, and that forms a syntony. And we picked this candidate wheel6 because it was in the syntonic locus. And our question was that can it be a regulator of its downstream gene? Now first we did the expression analysis by RT-PCR to validate the RNA sequencing, and followed by checking for the expression levels across developmental stages, in which we saw that it's expressed mostly in the primary or the early larval stages. And we also did the ribosomal pull down to show that the RNA is only present in the total RNA, which is the lysate input, and not in the ribosomal pull down, which is the RPS12 pull down here. So you can see that the candidate was present only in the lysate input and not in the ribosomal pull down. These were the protein coding controls. And this showed that it was truly non-coding. Then we looked for the subcellular localization to find out what could its potential function be. We did isolate 3 fractions cytosolic nucleoplasmic chromatin, and we did a Western blot based validation. And then by RT-PCR showed that it is chromatin enriched. So this could suggest that it is epigenetically regulating or post-transcriptally regulating these other genes, whatever its function is. Now first we went for a morpholinear mediated targeting of this gene to identify its function. When we injected the morpholinear against this gene, we found that there were vascular defects. And further we also saw that there was cranial defects. So what you are seeing here is the head of the zebrafish, and these are the eyes on the both sides. The green labels the endothelial cells and the red labels the blood cells. So when we injected the morpholinear, it led to vascular defects and caused hemorrhage. We also then used a TALEN-based approach, which is a more stable approach, and when we injected, we found that it was again causing a pooling of blood, and we then went ahead and also tested this genetically to make sure that it's a true model. Now what we observed was that when we edited this line, there was a transient change in this phenotype. So we then went ahead and did a confocal based imaging to see this. If you see here, this is where the hemorrhage is, and you can see this clearer in this, and you can see that this hemorrhage gets cleared over time. So what we did was that we went ahead and did the whole transcriptomics on these animals, and we selected certain genes for the validation. And from here we could identify a gene ROBO1, which is going down in the hemorrhage showing animals, and it was going back up in the animals which cleared the hemorrhage. So we look back at what could this gene be doing. It is already known to be present in the brain, and it is associated with the cardiac function. It is known in angiogenesis, and more importantly, even in mice, when this pathway was disrupted, it was leading to a cranial hemorrhage. So this makes us feel that V6 is somehow a regulator of the ROBO1 gene. Now we also looked at how to identify the conservation. Like I said in the first slide itself, that these LHRNA genes are poorly conserved. So we found a conserved sequence which is upstream to it, and using CRISPR-Cas9 we edited this locus, and we found that the same phenotype was recapitulated. So this suggests that location could be conserved, and our gene is also conserved. So this is a summary of the work. We identified chromatin enriched LHCRNA. When targeted by morpholina or talin, it led to a loss of vascular integrity. When we did a whole transcriptomics, we identified the gene ROBO1, which could be going down, which could suggest that that's why we get a phenotype. And when we edit a conserved sequence upstream, we recapitulate the phenotype, which means that this gene could be conserved. So with that, I acknowledge all the people. It takes a village to get a PhD, and this is my boss, Dr. Sridhar Shivasubbu. He has given me a lot of freedom to do this work. These are my DAC members, Dr. Vinod Skaria, Dr. Mitali Mukherjee, and Dr. Shantanu Chaudhary. All of their comments have been very useful for my work. These are all the team behind us, all of us working. And I want to thank Google, PubMed, and Sci-Hub specially for my work. And questions? It will come at the end. Yes. I had to go a bit fast, but if anybody has questions, I'll be happy to take.
Physiology of the cells that might actually leads to change in your RNA profile?
So I think when we do all these transcriptomics, we also look at the genes which are upregulated and downregulated. So when we do the G annotations, we'll be able to see if there is a huge change in the physiology of the cells. Probably we should be able to see that even in the pathways. So we don't see that, so we go ahead with those experiments.
Also, there are lots of methods to isolate RNA from a specific cell type without actually separating the cells from its surrounding environment.
Yes, but there are a lot of limitations to each technique. This is possible in our lab, and we went ahead with that. Thank you. Any other questions? Okay. So thank you, and I am active on Twitter if anybody wants to connect. Like Dr. Shilpi said. Thanks again for the opportunity.
Thank you so much.